All right, in this video, I'm going to be going over the 2026 AP Biology exam FRQs that were just released. And what's different about this video than probably just looking it up on your own is that I'm going to try as an AP Biology teacher to explain it to you. So regardless if you just took the exam or if you are using this exam as a preparation for your upcoming exam, I really hope that this is helpful. Once again, these answers in this video are not official, and these are just my predictions. So we will wait for the AP actual scoring guidelines to come out in July 2026. And when they do, I'm going to link them down below. So go ahead and take a look at that as well. If you want to follow along, I've already linked the FRQ, the actual release FRQ PDF down below as well. So you can go ahead and use that as a resource. All right, so we got timestamps. I'm going to go ahead and add timestamps. So if you want to go ahead to any particular question, you can go ahead and do that. And once again, this video was produced the day the FRQs were released. Okay, so the scoring guidelines usually come out about 2 months after when scores come out. So these are just my guesses. So go easy, and if I got something wrong, go ahead and comment below. That's very helpful to the viewers. And yeah, once again, I'm not a YouTuber. I'm a teacher, and every year when the FRQs get released, I personally like to sit down and do them. I set a timer for 90 minutes and I take the FRQs just like my students did. And it's good practice for me to reflect on and to improve as an educator. So yeah, hopefully this is helpful to you. This is our second year, I believe, doing the the AP Biology exam as a hybrid exam. And what that is all multiple choice will be done on students' laptops, and the FRQs will be shown in the Bluebook testing app on the laptops, and students will complete their responses in the free response booklets provided by the College Board for the exam. If you've never seen an the Bluebook app, here's kind of what it looks like. Here's a sample that I found online, and that was shared with me actually. And on top, there's a timer. There's a countdown timer, and that is helpful. All right, so here we see that all teachers are able to discuss APFRQ potential answers with students 2 days after the exam. And because it's 2 days after the exam, technically, hopefully I can do that. And this is legal. All right, here we go. So as we can see here, the timing on the FRQs. Typically, we recommend, because the first two FRQs are 50% of the score and the last four are 50% of the score. Typically, you want to break down the time pretty evenly. Now, for me, historically, when I've taken these AP FRQs, what I've noticed is that I've taken more time on the first two FRQs than the last four. So it all depends, but you really want to keep track because there's easy points, in my opinion, for the last four FRQs versus just the first two FRQs. I've did a quick count, and there's 16 total parts or questions in the first two FRQs and 16 total parts in the last four FRQs. So overall, there's about 32 questions on the 2026 AP FRQ. All right, here we go. So let's start with FRQ1. This one took me, I believe, the longest over all of them. Actually, 1 and 2 took pretty long for me as I was going through it. Let's begin. It says molecules known as dinucleoside polyphosphates are a type of nucleotide that are signaling molecules that accumulate in plant cells during dry or stressful conditions. So you don't want to spend too much time on the first question of these FRQs because they're, in my opinion, gimme questions in terms of they're just easy points. So don't think too much when you're answering these first ones, because as the FRQ goes on, usually the last few questions in each FRQ are more challenging questions to get. So it says to describe the three different structural components of a nucleotide. And here's my quick response that I wrote out. A nucleotide is made up of a phosphate group, a nitrogenous base, that could be A, C, G, T, or U, depending on DNA or RNA, and then a 5-carbon sugar, which is ribose for RNA and deoxyribose for DNA. In my class, I teach my students an easy way to memorize the building block of a nucleic acid is PBS. Here, phosphate based in sugar. And here we see DNA because it's double-stranded, and we see that it's DNA because it's also involving a thymine. But one nucleotide would be one phosphate, which is the orange, one sugar, which is the deoxyribose sugar, that's blue, and then a base that's in the middle that's hydrogen bonded to the other side base. All right, so there's that. Okay, so hopefully that helps. Next, it gives you some reading. Now, for each of these readings, for me, I read it one time. And as I read it, I quickly write notes on the side as I'm taking this with paper and pencil. So um, if so I wrote down some notes at the bottom, so hopefully that kind of explains to you what's going on in my mind as an AP Bio teacher as I'm reading the AP FRQs. I'm going to read this out for you. If you want to skip ahead, you can, but go ahead and follow along if you want. Stomata are pores on the surface of leaves that open when exposed to light, and they regulate the flow of carbon dioxide, oxygen, water vapor in and out of the plant. And in response to dry environmental conditions, many plants close their stomata, so I think of canned plants, which reduce water loss. Scientists hypothesize that the dinucleoside polyphosphates, and we've never, in my class, we've never learned these particular polyphosphates, AP4A, and here we see CP4C. They bind to this receptor called DORN1 on the plasma membrane of plant cells, and they initiate signaling cascades that cause stomata to close. The scientists expose plants to light to open the stomata and then place samples of the leaves in buffer alone or in buffer containing these three different compounds, AP4A, CP4C, or abscisic acid, ABA, a molecule that causes the stomata to close. The scientists measured so what's the dependent variable here? They took a look at um, the size of the stomata in each sample and calculated their sizes relative to the size of the stomata in the sample kept in buffer alone. All right, so there's the reading. All right, in addition, we see this graph that's given to us. And whenever I see a graph, I just quickly look at trends. Most likely you on your AP exam would have never seen this graph before. So you just got to kind of make sense of it and look for trends and which one's highest, which one's lowest, and the changes. Here we see that the buffer has the highest stomata size. And then these three that I just explained are pretty even, with ABA being a little bit higher, but not, in my opinion, that much higher, because if you look at the error bars, there's overlap. Okay, so that's what's going on in my head as I'm looking at this graph. For 1B, it says to identify the dependent variable, and we just did. I just said a little bit earlier that the scientists are measuring this based on the size of the stomata. So my quick response, and once again, you want to go quick on these beginning ones because look, if you're given 5 hours to do these FRQs, you probably do really well, but you're given 90 minutes. So it's a time test. So those who are able to quickly read and write down the answer, the better. You don't want to go too long in these responses, especially when it says to identify. So the dependent variable is the relative size of the stomata. That's my best guess on the answer. Here we see that based on figure 1, describe the relative size of the stomata in the CP4C treatment group as compared to the size in the buffer alone. And here we can quickly see that this is way lower than this one right here. So here's my quick response. In figure 1, the stomata in the CP4C treatment group had a relative size of, and I'm gonna go back, 0.6 is what I quickly calculated right there, compared to about 1 as seen for the size of the stomata in the group tested with buffer alone. Next, it says to justify. And once again, if you're just taking AP biology, to justify is to do a little bit more than identify, but to provide evidence, to provide data or scientific reasoning to support or defend or validate a claim. So you have to justify. So the question is to justify the use of buffer alone as a control. So if you've never learned, a negative control is usually what does not change, and a positive control is something that perhaps is proven that you are comparing it to, right? So the use of buffer one alone. So here's my quick answer. The samples of leaves in buffer alone served as a negative control in this experiment. So scientists can use this data to verify that observed results are due to the independent variable and not just based on other factors or errors or anything like that. So that is a great use of to be a negative control. In part C, so you got to keep moving on these FRQs. You got to keep moving because it's pretty long. As I showed you, it's about 9 questions in the first FRQ alone. Okay, so you got to keep going because every single question is worth good points. Part C, we see in a second experiment, the scientists used plants that were identical to those in the first experiment, except that the gene encoding DORN receptor 1 was mutated. The scientists repeated the procedure of the first experiment. The results are shown in figure 2. So I took some quick notes down here, and we know that all these will close the stomata. And those are some quick notes that I had earlier as I was going through it. Okay, so here is a different graph, figure 2. And in this one, we're seeing, let me just make sure this is shown here. Okay, so there's that. So here's the question. Justify the scientists treating one sample of leaves with ABA in these experiments. Okay, once again, ABA, if you forgot, is when that is added, then the stomata will close. So here's my quick answer for 1CI. In this experiment, ABA serves as a positive control. Well, why do we know that? Because earlier we learned that this acid is a molecule that causes stomata to close. So this proven outcome, this is a positive control, confirms that the plant samples are healthy, and thus they're able to close their stomata. So you want to include positive controls in good experiments so that you can go ahead and compare it to that as well. For 2, it says that based on figure 2, which is shown below right here, and I've screenshotted it right here, describe the difference between the effects of AP4A and CB4C treatments on the size of the stomata plant. So this is a simple question. It's just asking you to describe the difference between the two, and here's my quick response. In plants with mutated DORN1 receptors, we see the closure in both, with APA4A having about 1.1, I'm going to go back, about 1.1 relative size, and CP4C having about 0.7, as you can see. And that's all the question's asking. You don't need to explain why. It says to describe the difference between the two. For number 1C3, it says, here's the question. Activation of DOR1 receptors induces plant cells to produce a certain molecules that causes stomata to close. Based on the data in figures 1 and 2, if for cells treated with APA4A, predict, here's the question, the relative production of the molecules of DORN1 mutated cells in comparison to the production of non-mutated cells. Now, I want to go back here a little bit, because earlier it showed in the reading, and I'm going to go back and I'm going to box this, that both of these bind to DORN1 receptors on the plasma membrane of plant cells, and they initiate signaling cascades. If you don't know, in general, in chapter 11 of our book, we learn about cell communication, receptor transduction and response. So in order to initiate these signaling cascades, you have to have something bind to these receptors, and that causes these stomata to close. So here I've got it. Okay, so this one was a little bit trickier. I'm going to quickly try to explain this to you. So here I've highlighted APA4A in both figure 1 and 2, as it asks you to compare and to predict the relative production of the molecules. Now, this is not asking you to predict the trend here, for example, where this is in figure 1 and this is in figure 2. That's not asking that. Look back at the question. It says to predict the relative production of the molecules. So some of you may be thinking, what does that even mean, the molecules? Well, slow down here. Figure 1 shows you a normal plant. Figure 2 shows you a mutated plant. Okay, first understand that. Normal, mutated. Next, if you take a look at figure 1, really what is happening here is that the stomatas are closing, as we know in normal plants. But why are the stomatas closing? Because ultimately the plants are producing these molecules, these closing molecules. Okay, and here we see the trend here. Versus here, we see that the stomata stay open in the mutated plant. Well, why did they stay open? Well, once again, going back to the reading and understanding this experiment, it's because the plant did not produce these closing molecules. So see, here we see a relationship between closing molecules and whether or not the stomata stays open. So here's my response. I tried to explain a little bit right before. Looking at figures 1 and 2, we see that the production of stomata closing molecules in DORN1 mutated cells treated with AP4A will be lower. And I'm going to go back here. Will be lower than the production of closing molecules. That's not referring to this graph. Here you see the graph and you see that this is higher and this is lower. No, 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 no. We're talking about these closing molecules. The amount of closing molecules are going to be lower. Okay, so that's how I read the question. Could be wrong, but that's what I'm making sense of this. When you take a look at 1DI, here we see that the DORN1 receptor transmits signals that result in an increase of the transcription of genes involved in plant defenses. The scientists then developed DORN1 mutated plants in which the DORN1 receptor lacked most of its intracellular domain. Okay, here we go. In my class, we learned about intracellular receptors. My mind's already going there about how, in order to go through the cell membrane, you need to be small and nonpolar. and thus you need to do that to reach the intracellular receptor. So that's what's happening in my mind. Here's the question. In cells that are homozygous, some of you are thinking, how does that have anything to do with this mutation? I'll explain in a moment. Predict the effect of AP4A treatment on transcription, of the genes involved in plant defenses relative to the effect of AP4A non-mutated cells. In my class, we learned about testosterone and estrogen and how they're intracellular receptors and how they can ultimately change these transcription within genes. Okay, so here's some background on what I just said. Transcription of genes, intracellular scientists, they made intracellular. So ultimately, these scientists, this receptor is broken, but the outside receptor is still good. So even though you can get into the cell or send a communication into the cell, inside of the cell, the intracellular receptor is broken. Now, homozygous, what does that really mean? How I'm interpreting it, I could be wrong, is that homozygous means same. So both of the copies of this DORN1 gene are actually going to be broken, both of them, not just one of them. So here's my quick response. Hopefully this is somewhat making sense. If for D1DI, the cell will not increase the transcription of the genes involved. Now, what is the justification? Why is there not going to be an increase of the transcription? Here's my justification. Because signal transduction cannot happen. Because the intracellular receptor is broken, even if the ligand binds to the extracellular receptor that works, the message cannot be relayed to the nucleus to ultimately impact gene expression. So that's only FRQ1. So my big thing on when I'm taking this FRQ for 90 minutes, I keep thinking to myself, keep going, keep going, keep going, because every question is worth points and you want to move at a good pace. So you don't want to think too much about it. You just got to answer the question, move on. All right, here we go. FRQ number 2. Go ahead and take a look at this. All right, so this one was long and kind of challenging, but once again, pretty straightforward when the questions are given to you. All right, so let's go through this together. And it was long, and I will admit it was long. This one took me a while to get through as well. For FRQ2, it says that eukaryotic cells use siRNAs. Now, even if you haven't studied siRNAs or you never learned that in class, it tells you all that you really need to know. These are molecules that regulate the expression of certain genes by binding to these sequences in the target mRNAs. This binding causes the protein AGO2, which in my class we never teach, but once again, they tell you everything that you need to know in these FRQs. Now, this protein is important because it will cleave the target mRNAs. As a result of this cleavement, these mRNAs are not translated. Okay, so I wrote down some key things here. For part 8, you got to move quick. It's an easy question. Describe one location where ribosomes are found in eukaryotic cells. Here's my quick response. 2A. Within a cell, there are bound ribosomes that are located on the rough ER, and there are free ribosomes that are freely floating in the cytoplasm or cytosol. Same thing. Now, these proteins that are made in these two different locations are actually for different places, right? For within the cell or for within out of the cell, but it didn't ask for that. Okay, it gives you more information. Here we go. It's more fun. The following, okay, B, C, and D. I'm going to read this to you, so you can skip ahead if you just want the answer. To investigate the role of AGO2 in the cleavage of, and once again, cleavage of mRNA transcribed from the two genes, gene G and gene H, scientists determined the amount of these two genes, mRNA, produced in cells with 3 different genotypes. So here, I'm going to take some notes down here. Here you see plus, plus, minus, and minus, minus. Now, it kind of tells you that wild type is the most common, but more than that, the way I'm reading it, could be off, I could be wrong, but the way I'm reading it is that plus, plus means that there's 2 healthy copies of the gene. So I'm keeping that in my mind. Heterozygous plus negative means that there's 1 healthy copy of the gene, but it did say that it's non-functional. And then homozygous, which is same, negative, negative. In this way, I'm viewing it that there's no healthy copies of the gene. So I'm already thinking about that as I'm reading this. Here is some data that is given to us, gene G and gene H. Here we see 1, 0.9. So I'm already noticing a trend here that, whoa, that one's the highest. These start out about the same, but gene H goes, even though these are non-functional, no healthy copies of the gene, you're still seeing an increase here in the relative amounts of these mRNA. Okay, here we go. 2BI. So using the template, You're going to construct a bar graph. It tells you it's a bar graph. So here's a example that and that I'm providing you right here. And you know, you could draw it like this, or you could draw it with all three of the gene G's grouped together, and then all three of the gene H's graphed together. So I don't think there's one way or the other that's going to be correct. I think both are going to be correct as long as you graph them correctly based on what they gave you. Okay, so take a look at that. For B, the second part of B, based on the table, determine all the cells in which the amount of gene G mRNA produced is statistically the same. And as you can see here, it's for gene G, it's the filled in ones here. All of the error bars are overlapped. And in our class, we learned when you look at error bars, for gene G, all three cells mRNA levels are statistically the same. Why? Because there's overlap. Now, you may think that this one right here is just better, but so the error bars always tell us a different story. Because there's overlap, then the levels are statistically the same if you really think about it, right? Based on statistics. Part C for part one of C for FRQ2, based on the table, describe the relationship between the number of wild type copies of AGO2 gene and the amount of gene H in the mRNA of the cells. I'm gonna go back here. Here's some more notes. As I'm going through this, this is what I'm thinking in my mind. Gene G and gene H, it shows that the amounts that are produced. And once again, I told you healthy, not healthy, etc. Here's where my eyes are going, because the question asks, describe the relationship between the wild type and gene H. So my eyes are going here. I'm taking a look at 1, 2, and 3, it looks like, as it goes from plus, 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 minus, and minus, minus. Here's my answer. As the number of healthy AGO2 genes copies decrease. What we're noticing, okay, so what do you mean by decrease? Go. Plus, 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 minus, minus, minus. This one has 0 healthy. This one has plus. So as the number is decreasing, as the healthy genes are decreasing, I'm seeing that the gene H mRNA is increasing. Take a look. This goes from 1.0, 2.0, and 3.0. So that is how I interpreted that question. That's the relationship between the two, right? Here we take a look at C. Based on the data in the table for gene H, calculate the percent in which the average amount of mRNA in AGO2 minus minus cells increases compared with plus minus. Okay, percent. Now, here's where my eyes are going. Here we see from plus minus to minus minus. I see from 2 to 3. This is where my eyes are going on the data that's provided. Now, here's what's going on in my mind, and hopefully I can explain this to you, not just tell you the answer. So from 2 to 3 is a 1.0 increase. Now, if you think about it, 1.0 based on the start is 2.0. So that's half of your start. So if you're increasing by 1 and you're already at 2, then that would technically be a 50% increase. Not 100%. 100% would be up to a 4. And here we see from 2 to 3, not 2 to 4. So 2 to 3 would be, here's my answer, the average amount of mRNA in AGO2 cells increases by 50% compared with that in the plus minus cells. All right, that's my best guess for the answers. Let's keep going. Isn't this fun? Isn't this fun? I think it's fun. Here we go. Part D. Part 2. For this one, it says that anaphase 1 of meiosis, here's bringing in anaphase, is blocked in mice with AGO2, negative, negative genotype. Scientists hypothesize that the block in meiosis occurs because of the continued presence of a spindle fiber, stabilizing protein. Okay, it's talking about this protein now, preventing the chromosomes from separating. Based on the table, explain how the AGO2 negative, negative genotype could cause this effect on the dividing cells. So the claim is that scientists believe that this AGO2 protein is more important for controlling gene H than it is for gene G. That's the claim. Stay with me here. So here's where my eyes are going. Gene G, 1 to 1.5. Gene H, 1 to 3. Whoa, that tells you a story right there. And that's what I'm thinking about. Even though you don't know anything about AGO2, you can make that deduction right there. So once again, here's some notes that I'm just thinking in my head. The amount of gene H mRNA equals 3.0. Here, 1 second here. All right, it says the gene H mRNA is 3.0 versus 1.0. Once again, I showed you that a little bit earlier. That proves that gene H normally relies more heavily on AGO2 to keep its ultimate levels down. Okay, here's my best guess on the answer. The data supports the scientist's claim because when you remove AGO2, gene H, as I just showed you earlier, those levels are going to increase significantly, 3.0, while gene G only goes it goes from 1.0 to about 1.5, right? So that's what I would write there. For the second one, and here we go. Okay, got it. Okay, so then it says, so I just kind of explained the first one. Then it says for anaphase 1, whoops, I think I reread that a little bit. I think I read that wrong a little bit earlier. So yeah, so here's the anaphase 1. Sorry, I answered the first one first. The anaphase 1. Okay, so the problem is that anaphase is blocked, okay, in the negative genotype, and I'm getting all this information from the reading right here. Spindle fibers, as we know, they usually help divide chromosomes during meiosis, anaphase 1. AGO2 or siRNA, usually earlier in the text it said cleave the mRNA. Okay, so once again, I'm thinking in my head that the non-functional AGO2 protein is the negative. Now, there's a term that's produced right here, or that's shown here, called stabilizing protein. From what I'm gathering, and from what I just read right there, this more stabilizing protein is made. It's going to ultimately prevent chromosomes from separating in anaphase 1. Okay, so here's the best answer response that I can do for you. Because the AGO2 protein is missing, the cell can't remove the presence of spindle fiber stabilizing protein, which will ultimately stop these spindle fibers from separating chromosomes and dividing cells from meiosis. Okay. Now, I'm going to add to that because it's said here to, so in addition, based on the data given from the table, because you want to use the data that's given, gene H levels go higher, once again, 3.0 versus 1.5 when it comes to gene G. Okay, so that's my best guess for FRQ2. At this point, you're pretty tired in the FRQ. You got to keep going. I always think keep going. And don't let the time motivate you, but you just got to keep moving. FRQ3, we learned about cyanide, and this is only 4 points. So it's a little bit shorter than the first two, way shorter. So let's go quickly. Cyanide is a chemical that affects, and by the way, if you want, you can skip ahead, but I'm just going to read it out loud if you want to follow along. Cyanide is a chemical that affects the enzyme cytochrome C oxidase, CCO, a key enzyme in the mitochondrial electron transport chain. So we've studied that in class. Now at high cyanide levels, we're seeing that the activity is much lower than when no cyanide is present. At low concentrations, the cyanide CCO activity is slightly higher than when no cyanide is present, as shown in table 1. Okay, so here's some quick notes, and here's what's given to you. In my mind, I'm already thinking 2,234. Those are the numbers we used. I've never seen an AP exam actually ask you, because different textbooks say different numbers. So we use the number 38 in our class, 2,234. But I'm already thinking with oxygen, it's going to go through cellular respiration, ultimately going to the ATC and oxidative phosphorylation, chemiosmosis to produce 34 ATP. So I'm already keeping that in mind. Here's some more reading. So stay with me here. Some strains of bacteria produce cyanide, which they then release into their environment. Other strains of bacteria do not produce cyanide. Researchers grew both types of bacteria in high concentration nutrient medium and then isolated the medium from the bacteria. One half of each medium sample was left at high concentration, while the concentration of the other half was lowered. To study the effects of cyanide on mammalian cells, researchers then added 4 samples of media to the cells growing in culture dishes, as shown in table 2. They measured ATP and lactic acid production. So I'm already thinking, wait, when does lactic acid production happen? When you don't have oxygen and it's anaerobic and you only make 2 ATP. I'm already thinking about that. By the mammalian cells in each of the 4 treatment groups. Okay, so that's kind of what I talked about right there. All right, here's some more data that's given. 4 treatment groups, yes for cyanide, no for cyanide, high, low. Okay, so that's given to you there as well. So let's go ahead and answer that question. For FRQ3, Part A, describe the advantage to cells by producing ATP from one molecule of glucose by aerobic cellular respiration rather than glycolysis alone. I went really fast on this question because you don't want to waste time on these easier questions. Here's my answer. Aerobic cellular respiration with oxygen produces way more ATP. You don't even need to put a number. You can just put way more ATP per molecule of glucose compared to only using glycolysis. Now, if you do want to use the numbers, it's about 30 plus, or it's about 2 only. So it's like 15 times more when it comes to cellular respiration versus anaerobic respiration. Sorry, anaerobic, making energy anaerobically. Okay, not here. Identify 2 treatment groups that serve as controls. Well, when you take a look here, I'm already looking at something that doesn't add the cyanide, and that's 3 and 4. So my answer is really quick. The 2 control groups are mammalian cell groups, 3 and 4. Okay, because of the data that's provided. Okay, part C. 3C. Based on the information provided in both tables, predict the treatment group that is likely to have the highest ATP production for all 4 treatment groups. So I'm looking back at the information that's given, and I remember it said here that low cyanide will have slightly increasing CCO activity. So when I see this, I think of, okay, it's got to have the treatment group has to have had cyanide. So it has to be 1 or 2. But then which one is going to produce higher ATP? Well, go back. Low cyanide increases CCO activity. So in my opinion, it's 2 because 2 has both requirements. So here's my answer. The group with the highest ATP production will be mammalian cell treatment group 2. Part D, keep going, you can do it. The researchers claim that cells in which CCO activity is inhibited will show an increase in lactic acid production. Okay, I'm already thinking about lactic acid production, 2 ATP. Support the researchers' claim. Now, here's some quick notes. Normally, cells that use ETC, normally cells will use the ETC to make ATP, and that enzyme CCO is really important. Cyanide will block that electron transport chain. And when that happens, no oxidative phosphorylation will happen, but glycolysis can still happen with or without oxygen. All right, so there's that. So for D, what we're looking at is, here's my response. When CCO activity is inhibited because of the poison cyanide, the cell can no longer make energy using oxygen or respiration, but they can keep making some energy using fermentation, anaerobic fermentation for animals that creates lactic acid as a byproduct. Okay, so that's how I would support the researchers' claim. All right, here we go. FRQ4. Finish strong. So FRQ4, the process of meiosis enables animals that reproduce sexually to produce gametes. Okay, so here's a very easy question. Describe one characteristic of the chromosome movement during meiosis 1. Here's my answer. One characteristic of chromosome movement during meiosis 1 involves homologous chromosomes pairing up and then separating to opposite poles. The difference between meiosis and mitosis is that there's homologous chromosomes, one from each parent, that pair up during meiosis 1. Here's the part B. So you got to go quick on these easier questions. Don't spend too long explaining it. Just get the point, move on. FRQ4, part B, explain why chromosomes are more visible when viewed under a microscope during meiosis or during mitosis or meiosis than any other time in the cell cycle. The cell cycle is interphase, G1, S, and G2, and then the M phase, which is mitosis or meiosis. Now, here's my response. Here's my guess. DNA is typically in chromatin form during all of interphase, even during the S phase, until the start of mitosis or meiosis, when chromatin condenses into organized structures called chromosomes. And chromosomes are so much more easier to see than just a bunch of chromatin. For part C, it gives you a picture, and it says that an animal cell with 2 pairs of homologous chromosomes, typically number 4, has undergone meiosis to produce 4 gametes, and an error occurred in 1 step. Now, we learned that when errors happen, it's called non-disjunction, so that's already happening in my mind, as shown in the figure. 1 set of chromosomes is presented in white, one of them is presented in the darker color. Assume that each gamete produced by the cell in the figure is fertilized by the gamete with a complete haploid set of chromosomes. Also assume that gene regulation on chromosome 1 is not affected by the number of chromosomes, copies of chromosome 1. Predict the difference in mRNA production from genes on chromosome 1 among the 4 possible zygotes. Well, you can already kind of see what's happening here. None of the resulting gametes are going to be normal. Okay, and we've studied that in class. So here's my answer for 4C. The picture shows the result of non-disjunction, which occurs when homologous chromosomes fail to separate properly during meiosis 1. As a result, one gamete, egg or sperm, will receive more copies, so not a normal amount, but more copies of a chromosome. 4D, keep going. Triploidy, which is different than trisomy. Trisomy is when there's an additional chromosome, like we studied trisomy 21, trisomy 18. But triploidy is, it tells you right here, you don't even need to know it. Trisomy is a condition where an organism has an additional set of entire chromosomes, so 3 complete sets of chromosomes in each cell. Provide reasoning to justify the claim that most organisms with triploidy cannot produce gametes with the chromosome typical number of that in that species. Here's my best guess. 4D. Because organisms with triploidy have 3 sets of chromosomes during meiosis, these chromosomes cannot pair or segregate evenly into daughter cells, leading to gametes with unbalanced chromosome numbers. And that's not good when it comes to, or that's not preferred when it comes to humans or animals or anything like that. And it's usually non-viable. Keep going. FRQ5. Finish strong. Let's go. Lizards. throughout the Caribbean islands and Central and South America have pads of varying size on the bottom surfaces of their toes. If you want to read along, you can. If you want to skip ahead to the answer, you can. The sizes of the toe pads are genetically determined and not the result of phenotypic plasticity, okay, not based on environment or temperature. During storms with high winds, these lizards will cling to trees with the help of their toe pads. So cute. In recent years, an increase in both the number and intensity of storms have occurred across a range of these habitats. In 2017, there's 2 intense storms. 2019, the scientists investigate the relationship between the intense storms and the average toe pad size of the lizards that survive the storms. The scientists determined the average toe pad size of these surviving offspring and grouped their measurements into 3 categories: small, medium, and large. The scientists then compared the 2019 measurements with 2017. We're shown here 2 maps, 2017, 2019, and here you're also seeing greatest intensity of storm versus least, with smaller, medium, and large pad toe size. Here we go. So go quick in these first answers. FRQ5, Part A. Describe the role that changes in abiotic factors have in natural selection. Well, in our class, we learned that abiotic factors, such as weather, temperature, or natural disasters, can create a challenge for survival. Individuals with traits better suited to those conditions are more likely to survive. If you can survive, then you can reproduce, and ultimately you're going to pass on those favorable genes to impact the next generation. That's the definition there of natural selection. Survival of the fittest. 5B, based on the figure, explain the relationship between storm intensity and the average pad toe size. Here's my best guess. Larger toe pads can provide lizards with an advantage by increasing the surface area as they cling on trees, allowing them to survive in these high wind scenarios. These survivors will pass their genes or their alleles for large toe pads to the next generation, and ultimately you're going to see an increase of the frequency of the large toe size within this population as it continues. Part C, identify the region on the 2019 map by indicating the number that experienced the least storm intensity. Here we see least storm intensity. It's shown right there. And as you can see, here we see that according to the 2019 map, just to make sure. Got it. So based on what I can see here, Region 1 had the least storm intensity. I'm pretty sure I'm reading that right. Yeah, Region 1. Second guessing myself here. Hold on. When I took it, I put Region 1. Sorry, everyone. Okay. Is it? Okay. I'm going to stick to my answer. Pretty sure it's Region 1. Okay. But I may be wrong on that. All right. So let me know in the comments if it's a different region. Okay. But yeah, Region 1 had the least storm intensity. Okay. And that's what I first read it. Or that's what I answered when I took it. FRQ Part 5, Part D. Based on the information provided, explain how these continued differences in storm intensity can apply different selective pressures on these lizard populations living in the Caribbean islands, ultimately leading to speciation. How do you get speciation? Well, first, you usually need a geographic isolation, but that usually leads to reproductive isolation. And when 2 populations don't reproduce over or mix over a certain long period of time, then you can get speciation. So here's my best answer. Continued differences in storm intensity can cause populations to adapt more to their specific environment, which can lead to reproductive isolation and then ultimately lead to speciation. That's my best guess on that. Here we go. Last one, everybody. Finish strong. FRQ 6. All right. Raptors. Here. Raptors in Africa are top predators and keystone species in their ecosystems. Raptor population sizes have been declining over the years. Here's my notes on the bottom. Scientists studied the relationship between the conversion of raptors' natural habitat into farmland and the populations of raptors in those regions. They compiled raptor population size data from 3 different regions, region 1, 2, and 3. In each of these regions, scientists compared the annual percent change in the raptor population in areas that were protected from farming, and ones that were unprotected where farming happened. Okay, so there's some background there. Then you get a little box and whisker chart. And really what I read here is the difference between protected, as you can see in the clear boxes, versus the filled in boxes, which are unprotected. And already in my mind, I always look for trends. I see that this one's the highest. I see that this one's the lowest. Now, how do you read a box and whisker chart? Here you see the median, which is the line. Okay, so as you can see, this is the lowest median. This is the highest median. You can't see it right there, but it's 0. None are above 0 when it comes to annual percent change. So that's already what's happening in my mind. Here we go. Based on the box and whisker chart, identify the median percent annual percent change in population size in the unprotected area of region 2. So here's where my eyes are going. Region 2, unprotected. It's a darker one. And here is where I'm going all the way here. So based on the box, here's my answer. The median annual percent change in population size in the unprotected area of region 2 is, I calculate about 3%. That's what I looked at for the median. Okay, that's my best guess. For B, based on the box and whisker plot, identify the region and the area that experienced the greater median annual percent change in population size. So what I see here, the greater change, the greatest change, the one furthest from 0, is going to be this one down here, region 1 unprotected. That is my best guess that had the greatest median annual percent change, because if you look at the other ones, none of them went as far away from 0 than this one. Okay, that's my best guess. For C, almost done. Researchers hypothesize that if farming is eliminated in region 2, there will no longer be a decline in the raptor population size based on the data in the box and whisker plot. Evaluate the hypothesis. So take a look, region 2. This is if farming is eliminated. So is that true? Well, take a look. Here's my answer. The data does support the hypothesis. In region 2, the unprotected area has a median annual percent change of 3.0, indicating a decline, but the protected area has a 0%, as I just showed you. So this shows that if farming is eliminated in region 2, there will not be a decline in raptor population size. 6D, last one, come on. Based on the information provided, explain why ecosystem resilience in unprotected areas may decline over time. Here's my answer. Based on the graph provided, as I showed you earlier, we see that there is a consistent decline of raptors in unprotected areas. Ecosystem resilience in these areas may decline over time because of, here's my two thoughts. There's going to be a lower genetic diversity, or the loss of, if you think about it, a food web is made up of all the levels, but if you lose the top predators, it actually affects the overall ecosystem by a lot, especially if these are keystone species, like it mentioned. So it's going to be unstable, or it's going to collapse as a food web. Thank you. If you watched this video and it was helpful, I'm so thankful. I don't get any money from this. I don't get really anything from this other than the sheer satisfaction that people are helped and the practice that it helps me as an AP Bio teacher. So I hope this was helpful. I wish you the best on your AP test if you already took it. I wish you the best if you haven't taken it. Remember, you're not defined by your score. I know it's like the biggest thing you've ever done for those of you who are taking it at a very young age in high school, but you've got many, many more tests ahead. So regardless of how you did or how you will do, learn from it and just keep growing. Everyone has a great future ahead. I really believe that. I don't just say that. So keep going and have delayed gratification. Keep learning and enjoying biology. I love bio, and I hope you do too. And this is just one test of many, many. So do your best. Thank you so much for watching. And until next year, 2027.